All right, we've been talking lots about chromosomes and things, but now we're going to zoom in a lot closer and talk about DNA and RNA and nucleic acids and what they do and how we found out about them. So, in case you forgot, DNA stands for deoxyribonucleic acid, and hopefully you're aware that the genetic info is stored and transmitted from one generation to the next through DNA, okay? Really, viruses are the only things that use RNA. Everything else uses DNA for their genetic info. How did we figure this out? Well, actually, back in the day, people used to think it was proteins that were responsible for genetic info because of how incredibly useful proteins are. They do all of the things in all of the cells, and so it seemed like, well, they probably do this too. We didn't know much about nucleic acids, but there were 3 experiments in particular that helped us confirm that DNA was actually the source of genetic material and not proteins, which is what they thought. So the first one is Griffith's experiment, okay? And this had to deal with mice. So what you have to understand is Griffith took some bacteria, okay? And the one strain, the S strain, so here, right here, is these little dots here, these little like light dots, are little bacterias, okay? Notice they have this like dark orange little shell around them. That shell stands for smooth because this shell or this capsule, okay, kind of camouflages the bacteria so that the mouse's immune system can't detect it. It can't figure out that that's actually a bacterial cell, okay? So imagine you take this special S smooth bacteria, inject it into a mouse, mouse dies because its immune system can't detect it. Okay, that makes sense. Now you take some R is for rough because now the outside is bumpy. Okay, so you take this, you inject it into a mouse. The immune system can, with like cell-to-cell communication, it can directly feel that, oh, that's not from me. And so the immune system will attack and the mouse might get a little bit sick, but eventually the immune system will kill all the bacteria and so the mouse lives and will be healthy. Okay, then he also took some of this stuff right here, but he heated it up so it killed the bacteria. So he put dead, smooth bacteria in the mouse. Now, mouse is totally fine because the bacteria was dead. This all makes sense. These were all expected outcomes. Okay, so these were kind of the controls. We knew what was going to happen before it did it, and that's what happened. But here's where things get crazy. Okay, basically in this last one, they took these 2 and combined them. They took some of the living ones, which the immune system can detect and defend against, and they took some of the dead ones, which can't do any harm. They mixed it together, and we would expect the mouse to be healthy because individually they led to a healthy mouse, but when they were mixed together, boom, mouse died. What? And when they actually looked and took a sample, they found that somehow there were now these living cells. Living smooth cells. We didn't inject any living smooth cells. We injected dead smooth and live rough. What? So what they found is somehow the bacteria was transformed. This R rough bacteria was transformed into the smooth bacteria. So what was it? What was it that did the transforming? And here's where our next experiment comes in, okay? This one shows that it was DNA and not the proteins that did the transforming. So how did they do this? They took this mixture of with these heat-killed S cells, okay? And they took out lipids, they took out carbohydrates. So the only stuff in here is protein, RNA, and DNA, okay? And they like divided this big test tube into 3 different test tubes. In this one, they put in an enzyme that killed the protein. So what's left in here? RNA and DNA. They added in the rough cells. They've got heat-killed S cells. And guess what? We get transformation. Okay, in this one, they added an enzyme that kills an RNA. What's in here? DNA and protein. Okay, they add in the rough cells and these transformed cells appear. Okay, so both here we have transformation happening. Both of these had DNA in them. But in this cell, in this one, they put an enzyme that broke down all the DNA. So what's in here? Protein and RNA. And they added the R cells and none of those S cells appeared. No transformation occurred. So without DNA, there was no transformation. And the 2 that had DNA, there was transformation. And so that showed definitively that it must be DNA that's passing along and allowing this change to take place and not proteins. Because the ones where protein was in this one, nothing happened here. So that's how we prove that DNA was the source of info. And then there's one last experiment, okay? And this one had to do with viruses, okay? Viruses are basically two main things. They have an outer protein coating, and they have an inside piece of DNA. Some viruses have DNA, okay? And so what they did is they took something that was radioactive, and what a virus does is it injects its DNA into its host. Yeah, ew, okay? So what they did is they did two different experiments. In experiment one, they coated the outside protein with something that was like radioactive, okay? And so then this virus comes and infects this cell by leaving its DNA behind, and there was no radioactivity discovered, okay? Again, because the radioactivity was on the protein coat. So notice this virus takes its radioactive protein away. Then there was a radioactive source on the DNA. Virus comes, inserts its DNA in there, and now the radioactivity is inside the bacterium. So they can now tell by this tracer that, oh, it's the DNA that the virus leaves behind, and that's what infects the cells, not the protein. Again, so this is how we found out that proteins were not responsible, that DNA was. Okay, so let's go do a quick review of the structure of DNA. We've got a nucleotide. Here's a nucleotide right here. All DNA nucleotides have a deoxyribose sugar. It looks like this general shape. It's got a phosphate, and then it's got one of four nitrogen bases, A, T, G, or C. And so hopefully you remember that A goes with T and C goes with G. Okay. There's some additional vocab that you may or may not remember. There are the purines. This is adenine and guanine. You'll notice that they both have double rings. And the pyrimidines, which are thymine and cytosine, and those are single rings. Okay. A goes with T, C goes with G. One purine and one pyrimidine, 1 and 1, right? So they match up like that. The way that I remember the purines are as pure as gold, Ag, those are our double rings. And what would you rather have, a single ring or a double ring? I'd rather have a double ring made of gold, wouldn't you? Okay, so whatever helps, okay? And one thing to note too is that C and G have 3 hydrogen bonds in between them, shown by these dotted lines, and A and T only have 2. And so here's why we need one purine and one pyrimidine, is that if we had 2 purines and then the next rung we had 2 pyrimidines, our kind of like the rungs, if you will, right? Our horizontal parts of our double helix would be different lengths, and that's problematic. So we need one of each to keep the width constant. This was also backed up by some X-ray data. And so very, very quickly, I'm sure you've all heard of Watson and Crick, who are credited with discovering the structure of DNA. However, just like everything in science, they used the data produced by other people to figure this out. Those two people, Chargraph, figured out that C and G are always present in equal amounts, and A and T are always present in equal amounts. Interesting. And then this woman, Rosalind Franklin, was working, and she was taking X-ray images of DNA and found that they produced this type of image, which to scientists, they can look at this, and that helped them figure out that it was a double helix structure. Okay, and so without these pictures of Franklin's, these two dudes would have never been able to figure out the double helix. Okay, these two dudes got the Nobel Prize. She didn't. Partially that probably has to do that she was dead by the time they were given the prize, and also because she was a woman in science in the 50s, and you know, they were largely discredited oftentimes for their male counterparts. So that's kind of some of how we figured out that double helix structure. DNA is typically double-stranded. It has directionality. And what this means is we have the different ends. Whenever you have a strand of DNA, there's always going to be one side that ends with a phosphate group, and this side is called the 5' end. And there's always going to be one end that has an OH group on it, and that's going to be called the 3' end. And notice over here, 5' on top and 3' on bottom. And over here, 3' on top and 5' is on bottom. And that is what we mean by anti-parallel. Each strand points in an opposite direction. So this one goes 5 to 3, and then this one goes 3 to 5, the reverse. Okay? So DNA transmits genetic information, and the order of the A's, T's, C's, and G's is what gives the code, and that's what makes the differences in between us, okay? Different orders. And if the order gets changed, this is called a mutation. This can sometimes be good, sometimes be bad, or sometimes kind of just be nothing. RNA is very, very similar, except for it's typically single-stranded here. It has a different sugar. It has ribose instead of deoxyribose. It also has a phosphate, and it has a nitrogen base. C, G, and A are all the same, but U is different, okay? So T gets swapped out instead of U. There's also directionality with our 5' and 3', with the 5' being the phosphate group, okay? And so our base pairing rules are the same. C goes with G here, purine and pyrimidine, but instead of our T, we replace uracil. So there we go with that. RNA, what does it do? It helps make proteins. It helps with the structure of ribosomes, and it's also going to help us copy our DNA. RNA is also often the source of viral RNA, viral genetic information. So when we're doing our virus projects, you guys will learn more about that. And so basically looking at them, hopefully you can see they're pretty similar, right? They've got a sugar, a phosphate, and nitrogen base. And the backbones here, which is like the little ribbony looking part, that is formed by a covalent bond, okay, that makes this monomer here, makes it into a polymer. How are they different? Well, we've got some base pairing. This is how I remembered these rules. A 2-car garage and a used car garage. That's what I learned literally freshman year, and it still sticks with me. So use that if it's helpful for you. DNA has deoxyribose, RNA is ribose, and DNA is usually double-stranded, but there are some single-stranded kinds. And RNA is usually single, but can be double. The exceptions there are usually viruses, because they're crazy. So if you're wanting to see more information about this, check out chapter 13 in your textbook, and here are some of the main highlights from this lecture.